Randomly amplified polymorphic DNA, or RAPD, is usually found in the plural as RAPDs and is pronounced rapids, partly because it is a quick way to get a lot of information about the genes of an organism under investigation. The purpose of RAPDs is to test how closely related two organisms are. In practice, DNA samples from unknown organisms are compared with DNA from a previously characterized organism. For example, traces of blood from a crime scene may be compared to possible suspects, or disease-causing microorganisms may be related to known pathogens to help trace an epidemic. The principle of RAPDs is statistically based. Given any particular 5-base sequence, such as ACCGA, how often will this exact sequence appear in any random length of DNA? Since there are 4 different bases to choose from, 1 in every 4 to the power 5 stretches of 5 bases will, on average, be the chosen sequence. Any arbitrarily chosen 11-base sequence will be found once in approximately every 4 million bases. This is approximately the amount of DNA in a bacterial cell. In other words, any chosen 11-base sequence is expected to occur by chance once only in the entire bacterial genome. For higher organisms, with much more DNA per cell, a longer sequence would be needed for uniqueness. For RAPDs, the arbitrarily chosen sequence should be rare but not unique. PCR primers are made using the chosen sequence and a PCR reaction is run using the total DNA of the organism as a template. Every now and then a primer will find a correct match, purely by chance, on the template DNA. For PCR amplification to occur, there must be 2 such sites facing each other on opposite strands of the DNA. The sites must be no more than a few thousand bases apart for the reaction to work well. The likelihood of 2 correct matches in this arrangement is quite low. In practice, the length of the primers is chosen to give 5 to 10 PCR products. For higher organisms, primers of around 10 bases are typical. The bands from PCR are separated by gel electrophoresis to measure their sizes. The procedure is repeated several times with primers of different sequence. The result is a diagnostic pattern of bands that will vary in different organisms, depending on how closely they are related. Although we do not know in which particular genes the PCR bands originate, this does not matter in measuring relatedness. Diagnosis therefore relies on having a primer, or set of primers, that reliably give a band of a particular size with the target organism and give different bands with other organisms, even those closely related. RAPD results using such a primer are shown in figure. Grey mold, due to Botrytis cinerea, is one of the most destructive infections of strawberries and also attacks other plants. Classical diagnosis involves culturing the fungus on nutrient agar. It is slow and difficult due to the presence on the plants of other harmless fungi, which often grow faster in culture. As can be seen, RAPD analysis clearly identifies the pathogens from other related fungi, including other species from the genus Botrytis.